And I really found that he did pull stuff directly from the slides. And like this could be a really easy question that if you didn't really emphasize in your studying, you might have just like looked at this and guessed. So a PAMP, this is found on the pathogen. And what it does is it binds to the PRR that is on like your phagocytic cell. That stands for pattern recognition receptor. And once those two things like match up, they bind each other, whatever, then that phagocytic cell can like start, you know, phagocytosing the pathogen. So that was a really straightforward question, pretty easy. And this is directly from your slides. Well, if they're the same as ours, but it was right off the slides. In what area of the lymph node do B cells proliferate? So the answer here is going to be your germinal center in the lymph node. So B cells, they actually, I don't know if you can see this picture. So, oh, sheesh. Okay. Um, the B cell will migrate to the cortex of the lymph node, but where it proliferates, meaning like where you're going to like make more of these things, it's going to be in your germinal center. I also want you guys to really recognize this here, this T cell area. Um, so T cells and your dendritic cells are going to be in that area. Um, I'm pretty sure something like that had come up at a time or two. But so B cells will migrate to the cortex. In the meantime, they proliferate in this area. Note that the T cells populate the inner cortex. So I think it tricked some people on one of his quizzes or exams or something because they read like medullary sinus or medullary cortex or whatever like that. Not like they're not in the medullary sinus. The plasma cells are. Um, but the T cells and your dendritic cells will be in this inner cortex. So please recognize that. Sorry, I'm just checking the chat real quick. Is that called the paracortex or is that in a different organ? I think in
It
some
is.
things they
It is.
might call it the paracortex. Yeah, like Alex just said, it is. But he won't really use that word. He might use like medullary cortex if you see that. Because I think some people wanted to say like medullary cortex, but then they saw sinus. So medullary sinus will have B cells or antibodies. Medullary sinus, that, so what do plasma cells like make? Like how's the order here? Because antibodies come from what? Can somebody shout that out?
Plasma cells.
Plasma
Right,
cells.
right. So yeah, your plasma cells, they're going to be in the medullary sinus, and what they do is they make your antibodies, but you're not really going to have the antibodies like in there. The antibodies are going to be kind of floating around until, they find their antigen to like complex. Does that make sense? Okay, so question 3, patient comes into your office, presents with symptoms consistent with Neisseria infection. He tells you that he's been treated for one of these infections before. You tell him he may have a deficiency in what complement factor. So that's going to be a deficiency in C5. Um, so if I had listed C6, 7, 8, or 9, those could have been answers as well. Deficiencies in any of like the terminal complement factors, meaning C5 to 9, can lead to increased susceptibility, sorry, I spelled that wrong, to Neisseria infections. C5 to 9 are going to be your complement factors that create your membrane attack complex, specifically C5b. And then from there, you're going to have 6 and 7 bind, then 8, and then 9. In C9, you're going to end up with just like a lot of C9s. Like I know there's one slide where it says like C5b 6, 7, 8, 9 with like an N. And that means that you're just going to like be accumulating a bunch of C9s because that's the one that's going to put the pores in the membrane to allow that pathogen to be like killed. The most potent anaflatoxin is, and this is C5A. So your anaflatoxins, they are C3A, C4A, and C5A. In order of stability and potency, the most potent and stable is C5A, followed by C3A, and then C4A. Um, Anaflatoxins, so they're called anaflatoxins because what they do is they go recruit like neutrophils and stuff like that to mount an inflammatory response. Yeah, 534. What is C2B considered? C2B, um, it's not considered an anaflatoxin.
It's chemotaxis. It has a chemotaxis property.
Mhm.
Same
Um,
thing with C5A, but
Sorry guys, I'm trying to like check the chat at the same time. Yeah, we're gonna talk to Dr. Chusa, but like I said, I am totally not against you guys taking screenshots on your laptops or whatever you're zooming from, or just taking pictures with your phone. But typically, Chusa tells us not to send these out. I will ask her though. Um, but yeah, so what I want you to know with the anaphytoxins is the order of stability and potency, and then that they're called anaphytoxins because what they do is they induce inflammation, and they do that by recruiting your like inflammatory cells, aka your neutrophils. Question 5, you're working as a resident and rounding on the pediatric unit. You see a child come in and presents with teeth that look like fangs and other abnormalities in the development of tissues derived from ectoderm. You suspect an impairment in the activation of. So this could have been a tricky question. Um, so it's an impairment in the activation of NFKB, but IKK gamma is what like activates this. So you are lacking IKK gamma, but that leads to impairment in the activation of this, and that is describing Nemo deficiency. This is also called X-linked hypohidrotic ectodermal dysplasia. Um, we haven't covered NFKB. Are we responsible for those? So this was in chapter 3. I'm not sure how far you guys made it in chapter 3, but we were told that the quiz covered chapters 1 through 3. We didn't
He
cover like,
said we're responsible for everything 1 through 3, even if he didn't cover it.
and that's what I remember on our quiz. We had
Right.
covered most of chapter 3, but again, that's why I told you guys, like, I had focused super hard on chapter 3 because I know he didn't make it all the way through, and we were told that was on our quiz. But like I said, like, I remember there being a lot of chapter 1 on this first quiz. But this,
I named chapter one was heavy.
mhm.
It was basic on that aspect.
So if you didn't cover that, I'm sorry. Now you have a new fun fact. But it'll show up on your exam anyway. So, which inflammatory cytokine is responsible for recruiting neutrophils and guiding them to the infected tissue? This is CXCL8. It's a chemotactic factor, and what that does is it goes and grabs your neutrophils. And neutrophils are always going to be your first cell at the site of like injury. It's the starting cell of the inflammatory response, and after like usually 48 hours, like two-ish days, then that'll start being replaced by your macrophages. But the first thing to induce inflammation is going to be your neutrophil. This acts as an opsonin to trigger the classical complement pathway. That's going to be C-reactive protein. Um, Remember with complement, so there are 3 pathways uh, to like begin the whole complement cascade. You have your alternative. It's called alternative because it was discovered second, but it's usually the first to like act. And the alternative pathway is strictly just like C3 comes in there, and then you start accumulating C3b on the pathogen surface. Then you also have the classical complement pathway and the mannose binding lectin pathway. And those are just like, so classical can be started with either C reactive protein or an antibody. If it's going to be an antibody, it's going to be IgG or IgM. IgM is the stronger activator. So just keep that in your mind. And then also with complement, the alternative complement pathway, the convertase that you form there is C3BBB. The classical and MBL pathway both use the classical convertase, and that's C4B C2A. Um, those are also two little facts that I really want you guys to know. Question 8, all the following TLRs have their receptor located in the endosome except this is TLR4. This little chart here can be one of your best friends. He loves the TLRs. A lot of us miss questions on them, and he told us, you guys miss these questions. I'm going to keep putting them on your quizzes and exams. So just look over this chart. You don't need to memorize every single thing, but if you know where the receptor is, um, that's probably okay. And then also just which ones are for RNA viruses and stuff like that. So I'm checking the chat again. Does chromosome mean where these receptors are encoded? Also, wait, what did somebody need me to repeat something? What did you need me to repeat?
He was referring to the C3 convertases for the individual
Okay, so I'm just going to go
complement.
back to that real quick. Okay, so you have the convertases, and what the convertases do is they convert. So in the case of the alter alternative pathway, the alternate C3 convertase is C3BBB. And what that does is it converts more C3s into C3A and C3B. Because C3B is going to be the thing that binds to the pathogen surface. And the binding of C3B, that is the coding that's being done. So what opsonization is coating a pathogen surface so that it can be phagocytosed quicker and more effectively. So the convertase is just referring to you cutting your C3 into its respective parts. For the mannose binding lectin pathway and the classical pathway, your convertase is C4B C2A, and it does the same exact thing. It's still because both of those pathways still end up with you coding a pathogen in C3b. So it's converting C3 into C3a and C3b. It's just formed from those two other complement factors because it didn't start with C3, it started with C4. Does that make sense? And then the other question, for number 7, I thought it started with C1. So it does, but it's the part that's actually acting as the opsonin is C-reactive protein. I can show you guys that in the book if you want me to. Just remind me at the end. I'll attach um, the little clip from the book. Got it, thanks. Okay. And then what was that other question about a chromosome? I don't think I really understand what you're asking. Does chromosome mean where these receptors are encoded?
Yeah.
For the chart. Yeah. Sorry, I kind of got confused by your question. So with these, so you have, for example, TLR4. And the ligand, meaning what TLR4 is going to bind, is lipopolysaccharide. Um, it recognizes gram-negative bacteria. It carries the cells that carry receptors for these are going to be your macrophages, dendritic cells, mast cells, eosinophils. And then where that receptor is located on those cells is going to be in the plasma membrane. That's what this chart is saying. He asked a couple random questions about these, and he asked them throughout the semester too, because honestly, immunology really builds on top of everything. So things from chapters 1, 2, and 3 can come up later in the semester. So it's just good to keep those in the back of your mind. Is this chart in the lecture notes? Is this in chapter 3? I'm not sure. I I know it's in one of the study guides that I had sent you guys at the beginning of the semester. This is in the book. It might be from chapter 3. So again, if you guys haven't covered it, I would look it over before your quiz if he told you to know these things. Um, it is in the book somewhere. It should be in chapter 3. I don't know if he put it in your guys' PowerPoints though. And then this question, the feedback between macrophages and dendritic cells in infected tissue involves the secretion of what by the macrophage and what by the NK cell. So macrophages and NK cells have like a positive feedback mechanism. So macrophages secrete IL-12. IL-12 then goes and activates NK cells, and then NK cells activate or secrete interferon gamma, which goes back and induces more macrophages. So it's just a little positive feedback loop. And then I don't know if you guys really have gone over NK cells that in depth, but then it goes on to talk about the different types like CD56 light and dim and all that. But yeah, that again was just pulled directly from notes and out of the book. And then true or false, C3b binds non-covalently to surface components of pathogens. This is false, it binds covalently. I only threw this question in there because he doesn't typically ask things like this, but I remember there being like one random question about this. So that's just to kind of let you guys know that you want to like any picture that shows up in his PowerPoints. My advice to you and what I did was I would go to the book, find that figure, and copy and paste the caption from the figure onto my lecture notes. Because like I said, I really found his notes helpful for class, but like you at least if you're not going to read every single chapter, which I don't recommend that. I recommend reading the chapters, but if you're feeling like super pressed for time and you don't think you can get to the entire chapter, I would at least go look at the figure captions for whatever picture he has in his slides. And he puts the figure number on the bottom of every slide. If there's a picture on it, usually like in the lower corner of his slides, it's like figure 2.5. So I would at least go look at those if I were you. Can anything ever bind non-covalently to a pathogen surface? To be honest with you, I don't really know. Things do bind non-covalently, but I'm not sure if it's to a pathogen surface. I just know that he did ask like a really random weird question like this at one point, so just know those little minor details. And that's it for my section. Let me bring up Alex's answers.
On how I did the course, I thoroughly read the book. I read every chapter, took notes on the side. I thought the majority of what was in the book was what he said and what he tested on. So I lived and died by that. I know some other people did other things, but that's what I did. And especially for this first quiz, chapter 1, basic definitions were key.
Yeah, like you want to know clonal selection and the definition of that, like
just the basic spit back stuff will get you those easy points that, you know.
Okay, can you guys see this Word document? Or like what are
No,
you
it's a blank screen.
Okay. I'm sorry, I like don't know how to work Zoom still.
Oh, I have no clue either.
Okay.
All right.
I think you're able to see it now. Okay.
So the first one was just specifically what antibodies do. Quick spit back definition of neutralization and opsonization. No. But they're asking choose it for the person that just said that. So first one was just spit back what they do. I know that's just a very important thing that I know he loves going over. It went further in detail later, and it goes in a lot further in detail later in the course. But I remember for the first quiz, simple spit-back definitions were key of that, of opsonization, which is just the antibodies encoding the bacteria, getting engulfed in the macrophages. That's how it gets destroyed. And then neutralization is the toxins. Swollen lymph nodes, as we know, when you have to go through the adaptive immune system, they actually go back to the lymph nodes for proliferation from the engulfment of the dendritic cells, as we've said before, with the opsonization and everything, brings it back, undergoes clonal selection, clonal differentiation, all that other stuff. But this was just to show that you get the swollen lymph nodes from the actual activation of the lymphocytes from the draining of the infection site. The rest didn't make any sense at all. But that's an important, just to know the simple pathway of what happens of the innate immune system goes in, neutrophils go in, they attack it, not strong enough, dendritic cells come in, bring it down to the lymph nodes, clonal selection, clonal yada yada in the lymph. Gut tissue, you have to know just GALT, what are the components of it. You know that it's GI. There's also one in the lungs, but that's not really the main one. You got to know GALT and also know M cells. M cells are the actual lymph system in that area where they immediately slide through. There's a good piece written on that in the textbook. It was important, just the simple spit-back definition of that and what it is important. I know it was gone over in histology before, but there it is again. So the next one was just, again, spit-back definition of clonal selection and clonal expansion. Clonal selection is where you actually choose which one's the closest. Expansion is the actual differentiation and proliferation of it. You'll later on in the course, you'll get to how this actually goes on, but that's not going on right now. Just the simple spit-back definition of what happens is important. So that was
I would know clonal selection, like know the definition, kind of just like I think the definition is directly from the book. He asked probably like 2 or 3 questions on it.
Yeah, he has a good solid amount of things. Just the basic pathway initially, how this immune system works, was asked. Very broad based on that one. Highly recommend, if you read anything from the textbook, read chapter 1 thoroughly. That's a very good review of just the basics of what he can ask with PAMPs. That was very important before, in PRR. Very important. Know what they are, where they are, all that stuff. Vaccinations. He went along with this with smallpox and everything, with the woman getting scratched. Smallpox is important. But what did this go on? This told you that that a piece of the system was going on to activate the immune response of the adaptive immune system. Just to know basically what happens in the pathways, what happened with smallpox, what happened with the vaccine. Just the basic overlying fact. Someone ask a question? No? All right. So that's important, just the overview of that. Primary lymphoid organs, even back from histology, gonna be a spit-back question on every exam. What are they? Don't get confused with spleen. It's just gonna be asked everywhere, even on boards throughout everything. Cells, there's a good slide in the textbook that has all the cells listed out. I know we went over them again with the differentiation in class, but just the overview of what they are. There's going to be a couple basic questions. They're on every exam. There's going to be basic questions about cells. You should know them. Neutrophils, important to know about pus. So what happens is when you have an invader, neutrophil goes in. When neutrophils die off, who engulfs them? Macrophages. What do they appear like to the human eye? Pus. Just a common fact, gonna pop up. Straight definition on this one. The adaptive immune response includes all the following except C is part of the innate immune response. Even those simple questions are important. Know the difference between innate and adaptive. Chapter 1 stuff through and through. And definitions again, with edema is number 10. So that kind of stuff. But heavy hitter on chapter 1. Can't state that.
Real quick, do you guys, if you were to see a question that asked you which, like, immune, like, either innate or adaptive, which one was, like, stronger, which one would you say?
Adaptive.
Yeah, you guys should know that.
Which one's more selective?
Adaptive.
Adaptive.
Right, like, things like that will come
out?
up. And then also with, like, this first quiz, like, we're both saying it was very heavy, chapter 1 for us, which I've said multiple times, like, I didn't expect. That doesn't mean, like, only focus on chapter 1. We haven't seen the quiz. It's supposed to cover chapters 1 through 3. So even if you didn't get to stuff, make sure you go through chapter 3. Obviously he's going to ask you more of what you talked about, but, like, I don't want you guys, just because we're saying it was heavy on chapter 1 for us, to only focus on chapter 1.
Also, that chapters 3, 4 actually start the building of what he's going to ask on in the exam, which you have to know that information. Because chapter 1 is just the basic intro. 3 and 4, you actually start building. So keep up with it.
Do you guys have questions? Are there things you want clarification on? Are there questions that we wrote that you didn't understand? Do you know neutrophils present? Are you asking if they present, like,
MHC?
Can we go over toll-like receptors and interleukins? Okay, so to answer the neutrophils presenting stuff, neutrophils, their job is to go to the site of injury, and they're also phagocytic cells. So they start like the whole inflammatory response, but neutrophils die very quickly, and they are like, so you guys, I don't think went through, like, the NETs and stuff like that really that much yet. Maybe you have, but they release a lot of granules when they die, and those granules also keep killing the pathogen and stuff like that. And then neutrophils, they either just, like, kind of implode pretty much on themselves, or they're removed by macrophages, but they don't present anything. They just go to the site of infection and start killing.
And that's where it goes in with pus and where it comes from. Know that neutrophils are also in circulation, but they're triggered by chemotaxis, which we went before, which is either done by subendothelial injury. Again, back with what Holly went with before with CXCL8. That was, that's where it all comes back to, and that kind of stuff.
So I'm going to bring up the book real quick for the person who asked about TLRs. What specifically do you want to, like, know about them?
With regards to the CR1 CD35, I don't think for the quiz, but most entirely for the course. You should know whenever there's a CD or a CR or anything with the numbers and letters, he likes those questions and they're important.
Does anybody know the only complement receptor that's expressed on red blood cells?
Is it CR1?
It is CR1. That's a little fun fact that you should be aware of. I'm trying to find the TLRs.
I know, I killed this book.
What? Oh.
I killed this book back in the day.
What other questions do you guys have as I'm looking for TLRs?
Oh, and do the questions at the end of the book. The questions are very good at the end of each chapter.
Okay. So these are your toll-like receptors. So basically what toll-like receptors do is, okay, so they recognize like different things depending on which one that they are, and then they pretty much just like, hold on, let me find a better way to explain this. So, like this caption says here, they recognize like characteristics of whichever type of thing that they're going to attack. So some of them, like TLR3, they go after double-stranded DNA. Um, TLR8 goes after single-stranded RNA. So then they're going to bind to like the receptor, which again is in different areas depending on which TLR it is. So like some of them are plasma membrane, some of them are endosomes. Um, and then from there, they okay. Um, I like Alex, do you have input on this?
So when it comes to, first of all, yes, our questions are fairly close. When it comes
I
to,
work my questions around what I remember
yeah, same.
questions being like.
Um, so toll-like receptors important here with they're just your entrance into the cell and what it's going to
broke.
Go ahead, Alex.
and it's going what response it's going to trigger from that. So if some molecule like the ligand comes along and binds to it, it's going to initiate some sort of response. And you should know where it is and where it's found and what do they recognize. So that's where it goes into the ligands, the actual thing that's on it. What it oh my clicker doesn't go. The ligands, what actually is on it, the microorganisms recognize, so where it's actually found, what actually binds to that receptors, what does it do, like what cells actually have that receptor, and then where is it found. So that's where it goes from there, and all are used to signal some sort of response to the invader, whether it be a cytotoxic response, whether it be a humoral response, which it's discussed later in the chapters. But for that one, he doesn't go that far into when it comes to the responses. But you should know that, like, the basics of what goes on. So if an RNA virus comes, which of the following are used for it? Or NK cells used for RNA virus, which receptor? Like that kind of stuff. And if you look later in chapter 3, it actually goes along a whole pathway that ends with NKF beta, with signaling the genome changes, in that kind of respect. But I remember, especially on his examinations, that chart was golden for toll-like. I know he'll later discuss this in a bit more detail, but again, the book was golden for me when it came to that, but that and the pathway, which is stated later, which I do
If
not
you
have
know
on
this
here.
chart, like this chart would help you answer most of the questions that he would ever ask on toll-like receptors. I just remember him kind of like he emphasized this after our first quiz because a lot of people like missed questions on it, but they all could have been answered just based on this chart. Okay. Anything else?
What I also did was like online, or you can make yourself your own little chart with all the IL1s, IR so and so's, like all that stuff I kept and what they do, because that'll be really helpful when you study for the final. Or at least that's what I did. Did you use Janway's immunology? You mean this?
Somebody else asked me that question.
Is this Jan?
No,
this,
that's Cara. I
um,
don't even know what Janeway's is.
I don't know which analyzer it is.
I used
Was that the
this
book
book.
that he likes? Or he went
So
everything from this book.
I don't know. Yeah. This book is like where like if you're going to be reading chapters on immunology, this is the book you need to be reading. If
Yeah.
you want practice questions, the USMLE, the step 1 book that's specific to immuno, not like the first aid book. There's an actual immunology USMLE book. And those questions in there were very similar to the types of questions he asked. There's the BRS book. And then I know I sent you guys like just handouts that had practice questions on them. Those were good. Um, I even used the pretest microbio and immuno book that if you were in the January class, you took Indy already. Those were like good practice questions for Indy, but they were also really good for immuno. I did just about as many practice questions as you can. I'll tell you for every single class that I take. I think that's the best way I study. But for immuno, I found it to be really helpful because like things can be asked in many different ways. So the more exposure you get to practice questions, the better.
I thoroughly read for this course at least. And osmosis videos on YouTube are very good for like the basic components of everything. If you're just knowing where to start, those are very good. Oh, cool.
Okay, do you guys have any other questions? Again, like we're here, we can go for like as long as you want. If you have questions, clarifications, whatever.
Ollie,
Arrow.
I
Yeah.
was a little confused about what is a cytokine and what isn't a cytokine. I know are interleukins considered cytokines and complements as well?
So, hold on, I'm gonna go back to there's like the things with those are I want you to know the inflammatory cytokines, and that is gonna be like um IL-6 is an inflammatory cytokine. Like those are the ones that you should really be familiar with. Um,
That's
When
in
it,
the fourth section of chapter 3.
oh,
when it comes to, if it comes to what you're saying with definitions, cytokines are used for the inflammatory response plainly, or just it's like a general word. It matters on what subtype, like a chemokine is part of it. Um, like that's what it's going for. If you're meaning like toll-like receptors, is not a cytokine, but the ILs are right there. That image is a great one.
this is
TNF
the one
alpha
you should be
is
familiar with.
So, what about the complements? Are those considered cytokines?
no. Those
No.
are those are just proteins that
Okay.
help.
Yeah.
Antibodies are not cytokines. Um, but like how I always saw that, and that was just the numbers and the letters.
So, are cytokines just exclusively coming from the macrophages?
No, it can be sent from other cells too. Remember?
Okay, like
Like your
neutrophils
NK
or plasma
cell.
cells?
Remember NK cytokines with that exchange mechanism with NK and macrophages with 15 and 12. The question that we had before. That one is again, but in that aspect of like nitty gritty with what you're saying, um, that's not really going to be asked. It's mostly on what they actually do. You
Yeah,
know.
I kind of get confused when I'm reading it.
Yeah, don't get caught up in that detail. Get caught up in what they do. You know, and the exchange. I always thought of this at least in like a story kinda. Like I don't know, when I was studying I kind of just said it out loud all the way through and everything just kind of added on.
Thanks, Alex.
Now you're welcome. But
Okay.
read the book if you read chapter 1.
Okay, somebody just asked me to repeat the books that I used for practice questions. So I used this book here, the Param book, there's practice questions at the end of each chapter. The answers, I believe, are all the way in the back of the book. I used USMLE. Like it's specifically immunology. I'm gonna bring it up here to show you guys in case. I feel like a lot of you are thinking it's the first aid book. It's not the first aid book. I used pretest microbio. Can you guys see this?
Yeah.
Yeah.
So this is it's step 1, specifically immuno and microbio. I used
That's good. I never saw that.
um
Where
yeah,
was
you
that
can test
preview?
microbio, and there was one more microbio book I used. I don't remember the name of it. Okay,
The
so
BRS has good
you can't
questions.
see the
Yeah.
questions and answers in this book. So
Live and die for BRS for physiology. Live and die.
okay, so in this book it covers a lot of stuff first. I think I might have went too far. I believe immunos. So here's like I think how it's set up is it does the questions like at the complete end of the immuno section. I'm not sure. But like yeah, here's some of them. So around page like 100 something ish, there would be the questions and then the answers follow them. So
Lippincott
I hope whoever's having problems with that. You're welcome. Anything else?
I want to know how neuro went. If everyone's okay. I didn't get any feedback from it.
It was pretty straightforward. I mean,
Okay.
there were a couple of tracked questions that he got a lot of heat from, but I mean, I thought it was pretty straightforward.
Gonna happen, but okay. As long as you guys,
Overall, it went really well, and he was happy with the overall results of the class.
Oh, okay. That's a good average. It's a very good average. That's actually a very high average. It's a good one. So good job on that one. Yeah, neuro, not, I read the book, but it wasn't,
All right, any more questions or concerns?
oh, that's very good. I'm happy about it.
Okay, if you guys are missing any of these books, or if you just need extra study guides or something, you can let me know. If you need a book of any sort, I would rather you like text me a not NYCPM email. I think a lot of you have my number, or you can just like email me through NYCPM, but like give me a not NYCPM email. I think most of you should have these by now, but if not, let me know. And then, yeah, like I said, this first quiz, it wasn't.
It wasn't. You got to get used to his questions. You can hear his voice.
Also, one thing I will say about his questions, so he can have decently lengthy stems, but usually all you need to answer the question is going to be in the last sentence or two of the stems. So like I know a lot of people always felt kind of pressed for time with his quizzes and exams. So my advice, what I did with them was I would usually read the last sentence or the second to last sentence on first. And then if I still couldn't really answer the question from there, I'd go back and read the whole thing. But make sure you read every single answer choice because some of them can like look very appealing. But you have to like his question style is a little weird. He'll like give you a whole paragraph, but like all you needed was like 2 words from the end of the stem.
Sometimes the answers are blatant.
Yeah. But yeah, like I said, I feel like the questions that I wrote, even some of the ones that Alex wrote, they pretty similar to ones that like
or on the
you
quiz.
would give. And then yeah, just those extra fun facts like CR1 is the complement receptor on the red blood cell. Maybe you know that. Yeah. And then let me know if you need anything else. Okay. So if there are no more questions or concerns, then we will let you guys go.
Yeah.
Have
And
a
you
good
guys
weekend.
email just in case.
And yeah. So we will
All
talk
right.
to you guys afterward. Have a good weekend. Good luck.
Have a good one.
Have a good weekend.
Hope Brady loses.